Good morning. Today we'll study the structure of the DNA. We know that DNA is a long polymer of deoxyribonucleotides, and the length of the DNA is actually defined by the number of nucleotides that are present. So it's a long polymer of deoxyribonucleotides, and the number of nucleotides or for a pair of the nucleotide is referred to as the base pairs which are present in it. So in this presentation, we'll be studying the structure of the polynucleotide chain and the packaging of the DNA helix. So first of all, you should understand that what is DNA? DNA is a long polymer of deoxyribonucleotides, और उसकी जो लेंथ है वो एक्चुअली डिटर्मिन करती है द नंबर ऑफ न्यूक्लियोटाइड्स और अ पेयर ऑफ न्यूक्लियोटाइड्स विच आर रेफर्ड हियर विच आर प्रेजेंट इन इट. नाउ what is actually this? This is referred to as the base pairs which are present in it. So what are the base pairs which are present in this polymer of deoxyribonucleotide determines the structure of the DNA. For example, a bacteriophage known as Psi-174, उसके अंदर 5386 न्यूक्लियोटाइड्स होते हैं. बैक्टीरियोफाज लैम्डा के अंदर 48502 बेस पेयर्स होते हैं. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and a haploid content of human DNA has 3.3 into 10 to the power 9 base pairs of nucleotides. They are present in them. So let us discuss the structure of such a long polymer in detail. Now, structure of a polynucleotide chain. We know that DNA is a polynucleotide chain. Let us recapitulate the chemical structure of the polynucleotide chain. A nucleotide के 3 components होते हैं, ठीक है. नाइट्रोजनस बेस, अ पेंटोस शुगर, शुगर भी राइबोज होता है आरएनए में, एंड डीऑक्सीराइबोज डीएनए में होता है, और साथ में इनके एक फॉस्फेट ग्रुप होता है. So there are 2 types of nitrogenous bases. This is all you have done already in 11th class. So 2 types of nitrogenous bases are there. These are purines and pyrimidines. So purines or purines, whatever way you pronounce, so it has adenine and guanine. And pyrimidines, they have cytosine, uracil and thymine. They are present in the DNA. Uracil is present in the RNA in the place of thymine. So uracil is replacing thymine in the RNA. That you already know. Now, this is the structure of polynucleotide chain. Here you can see how the thymine and adenine, okay, they are joined together with the help of the hydrogen bonds. We can see here that a nitrogenous base is linked to the pentose sugar. So nitrogenous base is linked to the pentose sugar. Pentose sugar. Here you can see this pentose sugar. Okay, this is a pentose sugar, the ringed structure. So a nitrogenous base is linked to the pentose sugar through a glycosidic linkage to form a nucleoside. So this is forming a nucleoside. So nitrogenous base and the sugar, when they are linked together, they form a nucleoside, such as adenine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. So when a phosphate group is also linked, when the phosphate group is also linked to the 5-OH of a nucleoside through a phosphodiester linkage, the phosphodiester linkage, then a nucleotide or a deoxynucleotide, depending upon the type of the sugar present, is formed. So 2 nucleotides are linked to 3:5, the 3:5 direction phosphodiester linkage to form a dinucleotide, and more nucleotides can join in such a manner. So these nucleotides, they are joined in such a manner to form a polynucleotide chain. So a polymer thus formed has at one end a free phosphate, and at the 5' or a ribose sugar. So at the 5' they'll have this ribose sugar, which is referred to as the 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the ribose has a free The ribose has a free 3' end of the polynucleotide, and the backbone in a polynucleotide chain is formed due to sugar and phosphate. So sugar and phosphate, they are actually the backbone of this polynucleotide chain. Now the nitrogenous bases, they are linked to sugar and they project, and then they form this backbone. So this is the backbone of this polynucleotide chain, the sugar and phosphate. Now in the RNA, when we see in the RNA, what we find that every nucleotide residue has an additional group. Every nucleotide has an additional OH group that is present, and also in the RNA, the uracil is found in the place of thymine. That is 5-methyl uracil. That is another chemical name for thymine. Now DNA as an acidic substance is present in the nucleus. That was first identified by, now this is the structure of the DNA. We'll study in detail how it was being discovered, and later on its model was being there. So this is the double helical structure of the DNA, and these are the nitrogenous bases which are present that forms the nucleobasis of DNA. And here we can see that the uracil, the structure of the uracil is replaced in RNA. Now here you can see the brief discovery of the DNA. Now we see that DNA, it is an acidic substance and it is present in the nucleus, and it was first discovered by Friedrich Miescher. Now in 1869 he discovered it, and he named it as nuclein. So Friedrich Miescher discovered it for the first time, named it as nuclein. However, due to the technical limitations such as a long polymer and the iska structure समझने के लिए, it was not very clear for a very long time. So it was only in 1953 that Watson and Crick, they are familiar names to you, Watson and Crick, you can see their picture here. So Watson and Crick, the full name is James Watson and Francis Crick. They based on the X-ray diffraction data produced by Morris Wilkins and Rosalind Franklin. They proposed a very simple but famous double helical structure. इन्होंने actually double helical structure, along with the help of Morris Wilkins and Rosalind Franklin, they have explained this double helical structure of the DNA, which you have seen in the previous slide also. Now one of the very important point here to note is that the propagation was base pairing between the two strands of the polynucleotide chain, two strands of the polynucleotide chains and the base pairing. But this proposition was also based on the observations of Erwin Chargaff. Now Erwin Chargaff, that he said that the double stranded DNA, the ratios between the adenine and thymine and cytosine and guanine, they are constant and they equals 1. So that is the Chargaff's rule, that is the ratio between the adenine and thymine and guanine and cytosine are constant and they equals 1. So the base pairing, complementarity, parental DNA and the daughter DNA, they were being explained in the DNA replication. Now the base pairing here is a very unique property of the polynucleotide chain. So these bases, nitrogenous bases which are present, they pair up in a polynucleotide chain and they are said to be complementary to each other. Complementary means this base pairing is complementary, that is the adenine pairs up with thymine and cytosine with the guanine. That is complementarity of the base pairs. And therefore if the sequence of the bases is one strand is known, then the sequence of the other strand can also be predicted. And each strand from a DNA, let us call it as a parental strand. Okay, each strand of the DNA, if we call it as a parental strand, it acts as a template for the synthesis of the new strands. ठीक है, एक strand अगर parental strand बन जाता है तो उसके ऊपर एक नया strand form हो सकता है. And the two double stranded DNA let us call them as daughter DNA. जो नए strands बनते हैं उनको हम daughter DNA भी बोल सकते हैं. They produced would be identical to the parental DNA molecule, and because of this, the genetic implications of the structure of the DNA became very clear. So this is the parental DNA, and these are the daughter DNA, which can be formed during DNA replication. So we can also call it as old strand and the new strand. So this is the new strand which is formed on the old strand because of the complementarity of the base pairs. What is complementarity? That these base pairs, they are complementary to each other. Cytosine pairs with guanine and adenine pairs with thymine. Now, what are the salient features of the double helical structure of the DNA? First important, you can see the structure here. The first important thing is it is made up of the two polynucleotide. It is made up of two polynucleotide chains, and where the backbone is sugar and phosphate. So ये जो sugar और phosphate की backbone है, ये इसका first salient feature है, sugar and phosphate. They are the backbones. The two chains, they have anti-parallel polarity. Means one is going into the 3:5 direction and another is going into the 5:3 direction. So why 5:3 or 3:5? That we have discussed in the first slide, that how this is formed. 5:3 or 3:5 direction, they run anti-parallel to each other. Now another important feature is the base in the two strands are paired through hydrogen bonds forming base pairs. So whatever base pairs they are being formed, they are formed through the hydrogen bonds here. Adenine forms two hydrogen bonds with thymine from the opposite strand, and guanine is bonded with the cytosine with 3 hydrogen bonds. As a result, a purine always opposites a pyrimidine. So one purine is always pairs up with a pyrimidine. That you can see either with the double bond or with the triple bond. Just a second. So here you can see how they are pairing up. As a result, a purine always comes opposite to a pyrimidine, and as a result, it approximately uniform distance between the two strands of the helix. There is a uniform distance because of this pairing up of the hydrogen bonds. Now the two chains are coiled in a right-handed fashion. So you can see here the coiling. The coiling is always in the right-handed fashion, and the pitch of the helix is 3.4 nanometer. 3.4 nanometer, that is the pitch of the helix, and it is 1 billionth of the meter, that is 10 to the power 9 meters. So what is nanometer? A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter. And there is roughly 10 base pairs in each turn. यहाँ पे आप देखेंगे कि हर एक टर्न के अंदर एटलीस्ट 10 बेस पेयर्स होते हैं. And the distance between a base pair in a helix is approximately 0.34 nanometer. दो base pairs के बीच का जो distance है वो 0.34 nanometer होता है. So the plane of one base pair stalks over the other in a double helix, and this is in addition to the hydrogen bonds, and that refers to the stability of this helical structure. So plane of one base pair stalks over the other in a double helix. So this is how this plane of one base pair stalks over the another, and this is the reason for the stability of this DNA structure. I hope all the points are clear. You can make a note of all these points, and you can remember them for studying the structure of the DNA. Then when we compare the structure of purines and pyrimidines, can you find out why the distance between the polynucleotide chain in the DNA remains almost constant? इसका रीज़न ये है कि जो दो बेसिस हैं, बेसिस जो स्ट्रैंड्स डीएनए के स्ट्रैंड्स में होते हैं, दे आर जॉइंड थ्रू द हाइड्रोजन बॉन्ड्स एंड दे आर फॉर्मिंग द बेस पेयर्स. सो एडेनिन हमेशा दो हाइड्रोजन बॉन्ड्स के साथ थायमिन के साथ जुड़ा होता है, एंड ऑपोजिट स्ट्रैंड्स एंड वाइस वर्सा. एज अ रिजल्ट ऑफ दीज बॉन्ड्स, द डिस्टेंस बिटवीन द टू पॉलीन्यूक्लियोटाइड चेन्स ऑलवेज रिमेन्स कॉन्स्टेंट. कि ये जो इनकी जो बॉन्डिंग है उसकी वजह से उनका जो डिस्टेंस है वो हमेशा कॉन्स्टेंट रहता है. Now this double helix preposition structure of DNA और उसकी simplicity it explains the genetic implication, and it became very revolutionary in the field of science, biology especially. So Francis Crick, You can see the picture here. Francis Crick, he proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA, and then it